Okay, shall we start? So, welcome everyone to the Australia-New Zealand BioBridge workshop. So this is one of our many virtual workshops where we try and bring interesting topics of discussion to the community across the Tasman, and hopefully, connect people as well. So today's workshop is about gut health. And we've got 4 fantastic speakers, 2 from New Zealand and 2 from Australia, as we always do it. So for those of you who don't know who the Australia-New Zealand BioBridge is, it's an initiative that has been running since 2020. And I think I'm right. I forget the time because it's been quite some time we've set this up. And it's really to try and connect the health tech innovation ecosystem between Australia and New Zealand. So all the way from the research community, clinical, as well as businesses. So thank you very much for making time, and I'm sure you get a lot out of today's session. But before we start, how about everyone just putting your cameras on if you can, and we can take a photo. Ella, you'd be smarter than me on this. Would you mind?
Okay, everyone ready? Awesome, done. Thank you.
Thank you. Thank you very much. And lovely to see everyone's smiley faces. So what we'll do is we'll just get started. And I'll pass you over to Lance Chia, who's our co-lead of the Australia-New Zealand BioBridge from the Australian side. And Ella Dixon, whom you all have kind of interacted with one way or the other, is also a co-lead from the New Zealand side together with myself. So Lance, over to you.
Great. Thanks, Di. Thanks, Ella, and welcome everybody. Thanks for the intro to the BioBridge, Di. And just one thing that Di and I are quite passionate about, one of the things we focus on in these workshops in particular is connecting people. And we've got some examples where through these workshops, people who didn't know each other existed have some common interests, and we have at least one example where we end up with collaborative research and collaborative projects working on together. So if anyone here, whether you're a presenter or just participating in the workshop, wants to be connected to somebody at the end of it all, just reach out to Di and myself, and we can make that happen. But yeah, first up, thank you to our speakers. And the first speaker called agenda that we've got here is Dr. Jerry Zhu. Now, Jerry's got quite a long bio here, so I'm just going to pick out some bits here just to let you know about who Jerry is, and he's a great supporter of the work I do in Australia. So Dr. Jerry Zhu is a senior lecturer and clinical research fellow at the Translational Gastroenterology Laboratory in the School of Medicine at Western Sydney University. He has an emerging international reputation in the fields of GI health, medical device development, which I'm happy about, and clinical trials. Jerry was awarded his PhD in 2014 from the University of Sydney before he joined and led clinical research at the Translational Gastroenterology Lab at WSU. During this time, he's produced over 40 publications, developed active collaboration with national and international institutions, including the University of Edinburgh, University of Auckland, didn't know about that, Jerry, John Hunt Hospital, and Royal Prince Alfred Hospital in Australia. He also serves as a clinical fellow in the GI motility clinic at Blacktown and Camden Hospitals in Western and Southwestern Sydney, and roles within advocacy groups, industry partnerships, and founded his own company, Amazing Gut. Oh, Gut Tech. Gut Tech company, Amazing Gut. Might have to clarify the name of your company there, Jerry. But yeah, no, thank you, Joe, for coming, and I'll hand over to you.
Thank you for that, Lance. Let me just share my screen and So can you guys all see that? Yep. Okay, cool. Fantastic. Yeah, thank you so much for this opportunity to present some of the work we do for BioBridge. I think it's very exciting when we can showcase how we create innovation and kind of how we build this ecosystem to kind of help industry and clinicians create their own innovation, but also for us to kind of create innovations in terms of gut health and GI motility. So I'm from the GI motility unit, and what we do is we try and bring innovation in the field of gut healthcare. Now, this comes in many forms. It could be innovation from industry. Different industry partners, they can come to us and we can support them in terms of clinical trial and doing research. And it can also be from our own innovation. So actually creating innovation from our own experiences and from looking at those unmet clinical needs from our own clinics, and then bringing that back into the research labs and trying to kind of create our own different types of technology or medical devices, and then bringing that into the market. So a little bit about our team. It's composed of Associate Professor Vincent Ho and myself. So Vincent is the gastroenterologist. So he's a clinician who's based at Southwest Sydney Local Health District. And myself, I'm a researcher. So I've worked in clinical research for about 15 years, all the way from my PhD days, which was based in University of Sydney. We used to make this microarray. It was a microarray chip that would kind of help predict prognostic indicators for colorectal cancer. So that interest in translating medical research into the market and so that commercialization process, I got a glimpse of that so all the way back during my PhD days, and then we sort of continued on even after my PhD. So at the moment, we've got a whole host of different PhD students as well. They've all been working on different projects. And I think the thing I like about our team is that it's not just medical researchers. We have a lot of different specialties. So we have a student who's a biomedical engineer. We have another who's a neuroscientist, and we have another who's a statistician. So we have a lot of different components of research who kind of all go together to kind of create innovations, but having very different perspectives. So I think with that combination, we're able to problem solve quite effectively. So the other thing is looking at the different facilities. And I think it's having access to these different facilities that really makes us flexible and kind of being able to create innovation and sort of foster innovation. So the first thing is the clinics we have. So we've got 2 clinics. We have the Campbelltown Hospital outpatient clinic. That's where Vincent sees most of his patients for GI motility issues. And we have a dedicated GI motility clinic over at Camden Hospital. Now, I like the Camden Hospital clinic. It's got a small clinic, but it's dedicated to diagnosing and treating patients with GI motility issues. So it's a fully functioning public clinic. Patients come in, they get a whole host of different tests done, things like esophageal manometry, anal rectal manometry, pH testing, anal rectal biofeedback, so a whole host of motility diagnostic and kind of treatments. But what I really like about having these clinics is I go into these clinics and having that first hands-on experience and kind of that insight into looking at what problems there are in the clinic, and then trying to bring some of those problems back into our research facility to kind of address those unmet needs. Because it's quite different when you're sort of on the ground doing the diagnosis, doing the treatment. You can actually observe and you can see what the real problems are. I think that's the thing about innovation is really trying to address those unmet needs that the health providers have on the ground. The other facility we have is our research labs. So that's based at the Western Sydney Uni School of Medicine. It's not too far away from Campbelltown Hospital. It's next door. And that's where we do most of our basic science research. So things like food science, rheology, organ bath, pharmaceutical tests on animals and mice and things like that. That's where we do most of our basic research and kind of bench top research. Now, the other facility that we have access to is the clinical trial facility over at MacArthur Clinical School. That's also quite close to Campbelltown Hospital. And I think that facility is sort of dedicated for clinical trials, and it has a whole host of consultation rooms and receptionists where patients who are involved in clinical trials, they're able to go in and then they're able to be recruited and to run clinical trials. And it's really having access to that facility and having our own dedicated rooms in those facilities that we're able to partner up with industry and then run these big scale clinical trials that you'll see a little bit later on. And it's a really handy facility to have because it's away from the research facility and it's away from the clinic, but it has the same facilities that you need to actually run these clinical trials. So
Jerry, sorry to interrupt you.
Yeah.
We're seeing a big dark grey block on the right-hand side of your slides.
Right.
I'm not sure what it is, but now it's on the left-hand side, whatever it is. But just so you know, whatever's on your slides, we probably can't see it wherever those blocks are, whatever you just moved.
How about now?
Oh, yeah, that's better. And there's one on the bottom as well, across the bottom.
Okay. So yeah. So that was the bar for the Zoom thing. Is it better now?
Still there, smaller, but better than it was. Yep.
Okay, cool. Yeah, I had to minimize the block, but yeah. Okay, cool. So the other facility we have is the fecal transplant. So the fecal microbiota transplant center, and it's a center that we've created just last year. So we worked with Campbelltown private hospital to actually create a dedicated facility to do fecal microbiota transplant. And we're finding in a lot of research we're conducting that there's that link between the gut microbiome and health in the gut and in the brain and different aspects of health. So we've created this microbiome transplant center where we're able to do dedicated FMT research. And it's the only fecal microbiome transplant center in Australia that's primarily focused on sort of clinical trials and research. And some of the research we're working on at the moment is looking at the FMT, the efficacy of FMT in long-term IBS patients, and also looking at the effects of FMT in people with mild cognitive impairment. So people come in with mild cognitive impairment, we do FMT, and we follow those patients for several years to kind of see if that improves their mental health as well, because we know that link between microbiome and also the mental health. So the other voice that we really want to focus on is patients' advocacy. So we know we have the link to the hospitals and the clinicians, we have the link to industry. So the voice that's always been missing is the voice from the patients and the consumer groups. So here we work with quite a lot of different consumer groups, things like esophageal cancer and Rotary Health Australia as well. But there was one particular group that we were interested in. It's patients with gastroparesis and functional dyspepsia. Now, at the time, there wasn't a dedicated advocacy group for these patients, so we actually created our own one. So me, myself, and Vincent, and a few different patients that we know, we all came together and we created Gastroparesis Australia. So we have a huge Facebook group, and we have a lot of different patients in that group. So I think one of the advantages of working very closely with these patient advocacy groups is that we're able to get that consumer engagement and feedback directly from patients, and we're able to kind of showcase some of our newest research and clinical trials opportunities and really get those patients' feedback. They're able to see what kind of research we're doing, and we're able to get sort of some engagement or some feedback from them to kind of see if we're going down the right path or are we addressing the right issues that's really important to these patients. So I think that's really an important part that's been missing previously, but now we have that link and we have that really strong connection with our patients as well. So we sort of linked up all these different groups, from the health providers to the patients to industry partners. So now I want to kind of showcase a little bit about our clinical trial portfolio and sort of how we work with different groups, different industry groups in terms of doing clinical trials and then supporting them in kind of what kinds of research that they're able to provide. So one of our biggest industry partners is Atmo Biosciences. So I won't go into detail about how their capsule works because I'm sure Carl will go into a lot more detail about it. But essentially, we help them run the pilot trial and also the pivotal, international pivotal trial as well. We were the Australian site leads for that. And then some of the data that we were able to collect for that, it's ongoing to the FDA, and then it's going to regulatory assessment as we speak. And again, telemetry with Greg is another big industry partner that we work with. And it's a very exciting sort of technology, especially for our patients with gastroparesis. They're very excited about the ability to be able to map the gastric contractions and having this new technology, hopefully to improve the types of diagnosis and treatment as well for these patients. So I won't go into too much detail with telemetry. I'm sure Greg will go into more detail for that. And then the other aspect that we're really keen on is looking at different types of fibers. So fibers are really important as a prebiotic. And a particular type of fiber that we're interested in is low viscosity soluble fibers. Now, in this case, it's partially hydrolyzed guar gum. And what's special about these fibers is that they have all the characteristics of a regular fiber. You know, it's low GI, it's able to kind of help out people be more regular, it's able to reduce cholesterol. But in this case, with these low viscosity fibers, it's able to be very low viscosity. So when you mix it in with water, instead of forming a gel, it's actually just the same as water. It's got the same consistency as water. And what we wanted to find out was that what are the long-term benefits of these fiber, both in people with IBS and in particular with people with gastroparesis, because people with gastroparesis typically don't have fiber in their diet. They have to remove fiber because fiber will help, fiber is not going to help with the gastric emptying. But we wanted to find out if these low viscosity fibers are able to provide the benefits of a fiber diet, but also not increase any of their symptoms. So we actually partnered up with Nestle Health Science, and we ran a kind of a short-term trial. So it's only about 6 hours. So on these patients with gastroparesis, and we found was that it was able to lower their GI levels, lower their blood glucose levels after a glucose challenge, but they had no increases in their overall symptoms. So it's a really great fiber with a lot of potential for these patients with gastroparesis. And at the moment, we're doing a longer term trial. So it's over 3 months to kind of see what the long-term health benefits are for these patients with gastroparesis to be on this low viscosity fiber. So another trial that we're looking at in terms of probiotics and prebiotics is looking at baby formula. So Nutritica Australia, they've come to us to test out their new baby formula. It's called Neocate. And what this formula has, it's a amino acid-based formula for infants with cow's milk allergy. And then so with this formula, so what's special about it, it's got a prebiotic and it's got a probiotic. So it's got a special bacterial strain and it's got some prebiotic features in there as well. And we're doing a fairly large clinical trial to kind of test out the microbiome effects in these infants and to see if they're on a probiotic and a prebiotic diet, if it's able to both improve the gut microbiome, but also so reduce some of the symptoms they have with allergies. So things like cow's milk allergies and skin allergies, and then also kind of their GI symptoms as well. Now, the last thing, so the exciting thing is this company called Amazing Gut. So Amazing Gut, so we created Amazing Gut, Vincent and myself. We created Amazing Gut really to launch this technology about providing biofeedback to our patients. So anal rectal biofeedback is a treatment. It's a well-recognized and effective treatment for people with defecation disorders. So things like chronic constipation or fecal incontinence. It's like physiotherapy, but for your butt, essentially. But the problem with biofeedback is that it's very time consuming, and a lot of patients in our clinics are not able to finish the whole course of treatment, or they're not even able to start their treatment because they don't have the transport or they don't have the time to go through the whole physiotherapy regime. So what we did is we create a medical device. It's a small little device that people can take home and use on the app. And on the app, it shows them how to do the exercises properly, and they can track their symptoms and their diets and things like that. So it's a very simple technology. And we created this company called Amazing Gut to try and launch this into the market. So it's quite exciting. So we finished a sort of round of pilot trials, and the trial data was very successful. And then we're in the process of trying to bring that into market. So I think a lot of experience we had working with some other medical device companies like Atmo, like Alimetry, has really helped us. It's sort of a learning experience that's helped us create our own company and see how far we can take our medical device as well. So so in that, thank you very much for hearing my presentation. And if you have any questions, just let me know, and here are some of my contact details as well.
Thanks very much for that, Jerry. I know we had to have a panel session at the end, but we always allow some time for some questions initially. So if there are some questions, I ask you to put your camera on and then ask your question to Jerry.
Could Diane, could you have a question?
Yeah, so Diane, had your hand up.
Oh, are you talking to me? No, I was just trying to give you a clap for your talk, actually.
Right.
So the
But,
hand waving icon, I think.
yeah. So there's Abbas as well, who's trying to wave to you. But actually, I do have a question. What's aside from what you've done so far, what's the advancement in the gut health innovation area, aside from what you've shown us?
Okay. Yeah, so I think with GI motility, it's traditionally, there hasn't been a lot of innovation around GI motility. You know, it's a part of GI health that's been neglected for quite a while, because it's very hard to measure, because it's a functional disorder. So there's no structural issues to measure.
Mm-hmm.
And I think, so what's really exciting is we're seeing a lot of technology, you know, both from Atmo and Alimetry and from us, using the latest technology to actually get new ways of diagnosing kind of functional issues. You know, because you can't see the issues, you have to be a bit creative. You know, you have to look at things like GI mapping, you have to look at things like transit time, you have to look at the gas profiles. So I think in terms of GI health, in the next few years, what you'll really be seeing is using some of these technology to be able to create, you know, very, very high resolution diagnostic profiles. And then using those profiles to influence how these patients are treated. So I think that's where the change is going to be. And it's obviously the other one that we're quite interested in is the gut microbiome. You know, everyone's talking about the gut microbiome in food science and health and everything. So that's why we've been doing a lot of work with the fecal transplant. It's not very pretty, you know, it's taking poo and putting it in a blender and then putting that into the patients. But with some of the results we're seeing, it's very, very exciting, because it's got a lot of, you know, potential in kind of not only changing gut health, but also mental health and overall health in patients as well. But I think we need to kind of be a bit careful, because nobody really knows how it works exactly yet. So I think that's where the more sort of basic science research is going to be coming in and kind of finding out more about that end.
Okay, thank you. I mean, that's really exciting.
Yeah,
that's the question I was going to ask, Jerry. That the FMT, like, really fascinating to me.
yeah.
And I hadn't even heard about the mental health application. I don't know, are there some outcomes that you can talk about already that you've seen?
Yeah, so at the moment it's still early days. So we have about 10 patients who have mild cognitive decline. So they don't have dementia or Alzheimer's yet, but they're sort of, normally these patients are at high risk of developing dementia. So we were able to capture them quite early on and then provide them with a FMT and then actually follow up. So at the moment we've only provided them with the FMT, and it's only been a few months. So we haven't seen any changes in their mental health yet, but we're hoping to follow them on for 3 years and then 5 years and then 10 years to see how many of them would develop dementia and how many of them would, you know, not. develop dementia. So we don't have any results yet because it's quite early days. But I think maybe I'll have something for you later on in maybe 5 years' time.
That's just a really interesting space. Any other questions from anybody?
Hello. Hi,
Hey,
sorry, I've got a
Abbas.
question. Thanks, Jerry. I've got a classic Abbas question, which is, are you embedding any economic evaluations alongside the clinical trials where you're collecting, say, EQ5D questionnaires to get an idea about quality of life changes over time in a structured manner?
Yeah, so that's a really good question. So the clinical trials with Atmo and Telemetry, they are collecting that data. It's not with our site, but I know they are collecting that data with some of the other sites. And for our own medical device, yes, we will be collecting the economical data. But first, we have collected the improvements to health, the quality of life questionnaires and things like that. But in the bigger trial that we're setting up now, the actual pivotal trial, will be collecting the economic data, because one of the advantages of a home-based medical device is the time you save. You don't need to take time off work, you don't need to get childcare, and you don't need to find a way to get into the clinic. But the actual treatment should be the same as the treatment you get in the clinic. So you don't see an actual improvement in treatment, but what you're saving is the time and the economics. So I think that's where we're going to be focusing a lot of the trial data, and then kind of using that to build a package to send off to the regulators. So yes, we'll be doing that in like larger trial.
Amazing. Thank you.
Thank you. Any other questions?
Thanks, Jerry. But if people do have other questions, can we have the panel session at the end where it's just almost open slather? So if people want to ask things then, or talk about what you're doing, which is relevant to the speaker topics, we're happy to hear about that too. So Di, I'll hand over to you to introduce the next speaker.
Sure. So everyone, I have the pleasure of introducing Peng Du. So Peng's an associate professor at the Auckland Bioengineering Institute, where Ella and I live as well. Now, the Bioengineering Institute is well known for its work around computational physiology and marrying that with instrumentation and experiments to really explain and understand the human body function. And Peng is one of our experts in the gastrointestinal area. His bio is in the agenda that Alice sent out, or is on the Eventbrite, so I won't read all of that. But Peng is a fantastic colleague, and he is very knowledgeable, as well as a very supportive of the whole ecosystem. So there you go, Peng. Over to you.
Awesome, Di. Thank you very much for that introduction. I hope everybody can hear me okay. I guess before I start, I'd like to give a shout out to Jerry, our first speaker, for helping us out with a PhD thesis examination. So there's a lot of support going on beyond this workshop. So it's great to see. Let me just share my slide. Here they are. Excellent. Can we all see it okay? Great. Okay, let's see. Okay, so Jerry's talk was very inspiring in the sense that he's actually, as a biomedical engineer, doing a lot of the commercialization clinical trials, beyond device development, actually putting these, trialing these products on actual patients. For my talk today, I think I'm going to step back a little bit to the basic science aspect of how we actually detect the electrical activation of the gastrointestinal tract, specifically the stomach. This has been the topic, at least being main topic of investigation by our research group at ABI for the past 15 years or so. So the crux of our research, and indeed a lot of our commercialization activities, is to identify the electrical activation of the stomach. and using that as a biomarker to gauge at gastric functions and diseases. I'm really glad Dai mentioned computational physiology, because on the left, you see an animation, a computer or mathematical model of the simulation of what we understand to be the sequence of activation of electrical activity in the human stomach. So what you can see there, the red is the depolarized or active region of the gut. You can see these depolarized activity basically propagates as bands down the stomach. And what actually happens if you stick a camera or endoscope inside the stomach is on the right. You could see that, the animation stopped playing, there it is. These electrical activities actually result in mechanical contractions. And depending on the state of the pyloric sphincter, that is the sphincter right at the end of the stomach, these contractions could either allow emptying of gastric contents or mixing of gastric contents. So still a very sophisticated and complex physiological process that we're still trying to understand. But for now, we're pretty happy to report that through years of research, we finally managed to map the sequence of activation in the stomach and have a pretty good understanding of how it relates to gastric motility. So where this is a problem is, as you can imagine, as is the case in the heart, when these electrical activities, especially in the stomach, become abnormal, they will impact the efficiency at which the stomach works. So on the left, you can see, again, another mathematical simulation of the normal sequence in the activation in the human stomach. But on the right, you can see the emergence of what we know as a reentrant activity. So these activities, again, much like in the heart, would invoke basically an ectopic activation of these events where it's not supposed to be. And indeed, if you can imagine these abnormal sequence, if they persist for quite a long period of time, they will disrupt the sequence at which the stomach would break down food or empty its contents, therefore resulting in a whole cascade of downstream problems in terms of nutrition and other significant symptoms. So we started our research about 15 years ago with Greg on really looking at translating these multi-array high-resolution mapping techniques to first animal, but then human patients. At the time, we worked with a collaborator that basically manually assembled these electrode plugs, and they were quite labor-intensive and often takes months of lead time. So colleagues at ABI, again, the cardiac colleagues, have basically developed these flexible polyamide-based electrodes, basically flexible circuits. They can be manufactured relatively easily and be tessellated into these patches and deployed on the surface of the gut. And of course, when I mean the surface, in this case, is the serosal surface that requires surgical access. So we first trialed these electrode array in animals just to test safety and placement. And then eventually we had the opportunity to test these recordings in human patients in OR. And what you see on the right are basically some sample signals of the human stomach electrical activation. So these little blips you see there are human gastric electrical activity going down the stomach. And the computational models you've seen earlier are basically created to recreate those sequences of activation. And the important thing that we discovered when we then took this electrode array in and applied them in gastroparetic patients is that in those patients, there were a high degree or prevalence of gastric dysrhythmias. So these are real activation maps with the red color showing early activation and the blue color showing late activation. So in the normal antegrade propagation on the left, the activity is going down from the top to the bottom of the stomach as indicated by the arrow. On the right is a classic example of ectopic activation where a new secondary pacemaker would emerge in the distal stomach. And this would result in clashing of these waves against the normal antegrade activity, and again, potentially reduce the efficiency at which the stomach operates. So what this series of studies really validated for us is that these gastric electrical events could be used as a biomarker for diseases. And these were also correlated to degradations or reduced cell counts of pacemaker cells in the gut. So there's actual cellular basis as to why these dysrhythmias or arrhythmias would occur. And of course, we didn't stay there with the invasive surgical access because those would be quite restrictive and limiting. So over the past 10 or so years, we've been trying to apply these multi-array mapping studies at various scales. So just going down the scale first, looking at the macroscopic scales, we can manufacture or use these macroelectrode arrays to really looking at the activation almost at a cellular level. But to apply these electrodes in a minimally invasive manner or non-invasive manner, we needed to reduce the invasiveness associated with the access of these techniques. So we first went to laparoscopic devices, and these could be delivered through a trocar, and then endoscopic devices, and of course, ultimately, we've achieved body surface mapping by placing an array of these electrodes on the body surface and record the resultant gastric electrical activity there. And this will be a topic that will be covered in depth by Greg in a later talk. But I think this is I like this diagram because it really shows, we start with quite an invasive technique, but gives very high quality data, but that has allowed us to establish the ground truth. But we didn't stop there and aimed to refine this technique by reduce its invasiveness, eventually achieving non-invasiveness, non-invasive recordings. So now I'd like to just switch gear a little bit and look at what I think is a potentially an exciting next new chapter in GI sensing. And that is related to conducting polymers. As some members of this audience will know, New Zealand has a very special connection with conducting polymers. The late Professor Alan McDermott was one of our latest Nobel Prize recipient. He was awarded the Nobel Prize for his work on conducting polymers. So these are basically, conducting polymers are basically semiconductors or insulators usually, but through a process called doping, the good kind of doping, you could actually make these semiconductors conduct. And it's been quite a hot topic recently or lately, especially in terms of wearables, because these things are highly biocompatible. They can be functionalized to record various biological signals, not limited to just the electrodes, and they're highly flexible, as you can see in the image here. So what we have developed at Auckland is it's an ability to basically 3D print these conducting polymers. This is quite a cost-efficient method rather than relying on high precision instruments. They're literally like printing, 3D printing conductive inks. And again, as I mentioned, these inks could be, I guess, changed to record different types of biological activities. And we have developed these techniques at the Department of Chemistry with an actuated robotic dispensing unit. Ultimately, our aim is to develop a multimodal sensor that we can put on the gut to record not just the electrical activity, but perhaps other significant biological signatures, such as molecules associated with microbiome, pH, temperature, or other types of neurotransmitters. You can see here we've done an initial animal validation of this array compared to the flexible printed circuit arrays we developed, and indeed we have shown that these conducting polymer arrays would have a improved signal-to-noise ratio when compared against existing polyamide-based arrays. Another thing I really wanted to show with this conducting polymer setup is its ability to stretch. As you can imagine, most wearable sensors these days don't stretch that well, because once you stretch the conducting track or the wire would actually break. And that is indeed the case under normal circumstances with these conducting polymers. You can see here, as we stretch, cracks in the tracks would appear. However, through some clever treatment of adding disorbitol as the additive in the process, we can see that we can actually make these stretch these electrode arrays, and the tracks wouldn't break. And in fact, would be well beyond 500% strain before we see any significant increase in the resistance of these arrays. So this would allow us to have a highly conformable and stretchable array that would perform independent of stretch in terms of wearable and implantable monitoring. So with that, I'd like to finish on this quote by one of the founding fathers in GI electrophysiology, a clinician called Walter Alvarez, all the way back in 1922. So that's almost a century ago, where he said gastroenterologists would use electrical measurements for routine diagnosis of stomach disorders, just as the heart specialist does. And of course, as we know, cardiology has developed leaps and bounds, and we're only catching up in the gut field. But I'm glad to say that we have made up a lot of ground in the past 15 years or so. With that, I'd like to thank you for your attention, and I'll be happy to answer any questions you may have. Cheers.
Thanks, Peng. It's always fascinating to see what you're up to next. I hadn't realized about the conducting polymer work, but that is actually absolutely exciting. Okay, are there any questions for Peng? Abbas, are you waving at us, or are you asking a question?
Just waving. Just waving.
Okay, just waving. Okay. Do we have any? So, Peng, can I ask, when do you think your conducting polymer technology is going to be ready to be moved out of the bench?
That's a great question. So this is why I think the 3D extrusion print or wet printing is quite good, because the requirement of manufacturing is quite low there. So it can be deployed in a cost-effective manner. Because currently, what people do is they require a lot of very high precision instruments to make these things reliably. So it's not very easy to commercialize these. So we're moving to a low-cost solution in an attempt to lower the barrier to entry.
Okay. I was just wondering whether there's a connection here with Tim Woodfield's work as well in Canterbury.
Well, I work with Ujranka at chemistry on this, but I'm aware of other other groups that are doing similar things in New Zealand as well. Yeah.
Cool. Okay. Any other questions? If not, we will.
Actually, a quick question there for Peng.
Okay, you've got a quick.
Gary, hi.
Hi, it's a great presentation. I was just wondering, because a lot of, I think at the moment, because we're measuring the gastric changes and like electrical signals there, but a lot of our patients have, similar issues all over the GI tract, with the esophagus and maybe kind of the lower gut. I know with gastroparesis, a lot of patients also have slow transit, sort of constipation. Can you apply this technology to other parts of the GI tract, or like, is it maybe there's just too much muscle and fat maybe for like the lower GI? Like, can you apply that to other parts?
There's no reason why we couldn't. I stuck to the stomach because it has the best signal out of all the gut. But yeah, I mean, other than access and, practicality of placing these sensors, there's no reason why they couldn't be applied anywhere else.
Okay, cool. That'd be good because what we do now in our clinic with esophageal manometry and anal rectal manometry, it's the same sort of data we're collecting, but we have to sort of, it's quite invasive, so you have to put a tube into their kind of GI tract, and some of the patients don't actually like that, and it sort of excludes some of the potential patients. So I think if you can put that anywhere on the GI tract, it'll be a huge plus for kind of diagnostics for motility.
Thank you.
Yep, it's great work.
Thank you.
Okay, so what we'll do is we can take any, and you guys can think about questions for Ping, and we can take that at the end of this of the panel discussion. Or panel presentation, sorry. Faye, would you mind muting yourself, please, because there's a bit of interference from your end?
Oh, sorry, Di, I didn't realize I wasn't muted.
That's okay. No worries, Faye. Thank you. Okay, Lance, over to you next.
Yep. Thanks, Di, and thanks for that, Ping. And to our next speaker, and we're very fortunate here to have Kyle. He's actually we've had a few references or mentions of the Atmo Biosciences technology. Kyle's actually co-inventor of the Atmo core technology, and he's managed many aspects of the project up to date, including running the human trials, but also the commercialization pathway. He holds a PhD in material science and electronics from RMIT University in Melbourne, authored over 35 journal papers, has 10 years experience in his PhD in postdoc work in developing biomed devices and running clinical trials, which kind of makes him a textbook candidate for the BioBridge activities that we get involved in. So thanks for coming along, Kyle, and I'll hand over to you.
Excellent, and thanks for having me present today. Just going to bring up my slides. So yeah, as you guys heard, I'm Dr. Kyle Berrien from Atmo Biosciences. I'm currently head of clinical affairs, but as you heard, I'm a co-inventor of the Atmo Gas Capsule and co-founder of the company. You also heard I don't have a medical background. I am an engineer and a material scientist by training. I did my PhD within the team that developed the Atmo Gas Capsule, and I led the first in human trial there at the university and led the commercialization of the IP out of the university, which, if anybody has tried to commercialize IP out of a university, can understand some of the struggles that I may have been through. But that's for a different presentation, and that's a much longer topic. But yeah, so I'm here to talk about Atmo Biosciences, what we're doing, and how we are commercializing our device. So when we started Atmo, and the idea is we saw gastroenterology and the field, as Jerry pointed out, of functional gut disorders as being stuck in the past. There's not a lot of great diagnostic tools out there. There's also a lot of dogma within the field. There's a lot of partisan camps where, people on, say, the East Coast of the US don't agree with people on the West Coast of the US. And, how are you meant to actually get to the bottom for the patient to actually get treatment outcomes for the patient? So we aimed to try to reduce some of these protracted timelines for patients to get better diagnoses and targeted treatments with personalized outcomes, whilst trying not to increase the current burden on the patient. So our overarching aim has always been, what if assessing GI health can be as simple as taking a pill? And we're trying to use gastrointestinal generated gases to do such things. So why are gastrointestinal gases important? Well, throughout the 9-odd meter long tube of the GI tract, you have multiple organs in here that have various functions. So if you're thinking proximally to distally, proximally in the stomach, the primary gases there are either swallowed or they're produced through chemical reactions. As you move distally through the GI tract, you get an increase in microbiota quantities, but also diversities. Now, these microbiota produce gas as they interact with the food that you give them, which has a direct impact on their host. And we can detect some of these types of gases throughout the gastrointestinal tract. And we can also detect where we are in the gastrointestinal tract. So we can tell if the transit through a specific organ is fast or slow. And, we've worked quite closely with Jerry. They've helped us out a lot in trying to understand this within pilot cohorts and within a pivotal study. And I'll go over that in a second. But realistically, what we're trying to do here is create a new field. Getting access to the outputs of the microbiota in situ is near impossible. So before us, there was no real way to understand what the gas environment is like in the GI tract. Because as soon as you try to look at it, you are disturbing the environment in which the microbiota operate in, and therefore you change its function. So the only way that they were really understanding this was through breath tests or through flatus measurements or through a fermentation of fecal matter. And that just gives you an indirect and not very accurate picture of what's going on throughout the GI tract. So we're trying to change that and add to a new field, which is quite a bit of work, but we have a direct path to market with a motility product, or just understanding of transit time, where we can gather all the information we need to then go on to other clinical indications. So our device, whilst the core component is the ingestible gas sensor capsule, we have a whole system that is integrated around it that consists of a number of different elements. So our capsule itself transmits wirelessly out of the body as it transits through. We have a receiver that is worn on the body by the patient that is able to collect that wireless data and store it for the duration of the study. Now, we do this because we want the patient to be able to go about their daily life with minimal disturbance to what they want to do. We've also developed what is the Atmo Motility Bar, or a muesli bar, which is a standardized meal. And when I was part of the core development team who invented the product, and when I started Atmo, I thought I would be making muesli bars, I would have laughed at you, but it is an essential part of running our study to try to minimize the impact of diet, specifically on understanding gastric emptying time. We also have a mobile device, or a tablet with an app on it, that allows the clinician to set up the patient before the test, and when the device is returned, it downloads the data from the receiver and uploads it to the cloud, where the clinician can access that data through a clinician portal, where they will review the results, generate reports, and make diagnoses. So as I mentioned, we have a clear path to market around motility disorders. One of the key reasons why this is chosen first path to market is that there is not only a predicate in the US that we are piggybacking off of, and that is the capsule there on the right, which is called the Smart Pill, which is from Medtronic, which gives us a very easy regulatory pathway for the US. But most importantly, there's a very clear CPT code, or a payment code, at the end of it within the US market. So Whilst we all think that, you know, just creating a great product that will help patients is enough, getting paid for the use of your device is actually quite important. Now, creating a new CPT code is a very long process and not something that you really want to be undertaking unless you're in a big company like J&J. So as a small company, you really need to try to go for pre-existing codes. Now, I'm really talking about the US. That is our primary market that we're going after first, and that is purely because it's an easier reg pathway, there's payment at the end of it, and it's a large market for us. Once we get into the US, we'll be looking at expanding out into other regions. So, which is unfortunate, you know, as an Australian company that we're not launching in Australia first, but it's just quite common. You go to the US first, bring it back. We are launching also in New Zealand because it's a low doc, low entry regulation country, which allows us to get some local runs on the board, I suppose. So our onboard sensors allow us to, as I mentioned earlier, measure anatomical locations, which allows us to output regional transit times. So these are gastric emptying time, small bowel transit time, colonic transit time, and whole gut transit time. So the clinical indications specifically that we are going after with these measurements are gastroparesis and slow transit constipation, and specifically how they relate or differentiate from other functional disorders such as functional dyspepsia. So currently, how do they diagnose these disorders? Well, gastroparesis is commonly, or the gold standard is diagnosed through gastric scintigraphy, which is a radio-labeled egg beater meal that the patient has to be at a radiology clinic for up to 6 hours. So there's radiation exposure, it's inconvenient for patients, it's time consuming for the radiology clinics, and also importantly, there's no revenue for gastroenterologists here. But importantly for us, it's only a single region of the GI tract that it generally measures. There are some places that do whole gut or multi-gut scintigraphy studies, but they're few and far between and have a significant time burden for the patient. To understand colonic transit time, or for slow transit constipation, they use radio-opaque markers, and these are just plastic pieces within a capsule that you take on multiple days, and then you take an X-ray on a few days after you've taken these capsules, and you physically count how many of these little pellets are left within the colon. So whilst it's considered the gold standard, it's obviously got radiation exposure. It's very inconvenient for the patient to have to come back to the radiology clinic multiple days in a row. It's again time consuming for radiology clinics to physically or manually count a lot of these white dots on the screen. And again, there's no revenue for gastroenterologists. And this is actually quite important because you want to make the gastroenterologists want to use your device, right? It's that they are running a business at the end of the day. They also want good patient outcomes, but they are running a business. And finally, quite often you see, and this is not just within motility, but this is throughout all functional disorders and GI, there's just a try and treat attitude here. There's poor diagnostic outcomes from a lot of the diagnostic tools or lack of access to diagnostic tools. So you know what? It's just easier to trial and error this. You go on symptoms, but the problem is these symptoms are overlapping and very non-specific. And so it does take quite a while for a lot of these patients to get a good outcome. So what do we do? Well, we measure, as I said before, multiple regions or multiple locations. So we measure ingestion, the gastroduodenal junction, the ileocecal junction, and body exit. Our primary outputs that are clinically indicated are gastric emptying time and colonic transit time. And within the pilot studies that I'm putting on the screen here, which Jerry did gather the data on our dysmotility patients there, we get very good diagnostic accuracy to the smart pill. And importantly, we detect multi-regional delay within a single test. Now, I don't have it in our slides here, but within our latest pivotal trial of over 200 patients, it's around a third of the patients only have a single organ delay. Most of our patients either have no delay or multi-organ delay. So understanding multi-regional delay is very important when you are trying to get a differential diagnosis and the right outcome for the patient. So just on our pivotal trial, this was a big undertaking for us out of Australia. We are lodging to the FDA very shortly, and we undertook over 200 patients within this trial. And as you can see from some of the logos on the screen, these are some of the most prestigious institutions involved in this in the trial, which shows that there's a lot of interest within devices like this because there's just really not a lot in this area. It also shows that we got over 200 patients in under 7 months of recruitment. And I think that was primarily driven because Western Sydney did such a great job in getting up and running quickly and recruiting really quickly that it got these other big names like University of Michigan and Mayo Clinic and Harvard kind of scared that they were going to be at the bottom of the author list, and they actually got their butts in gear and recruited quite well in the end as well. So we are just putting the publication of this trial together, and we are presenting it multiple times this year. But the trial was led by Professor Bill Shea out of University of Michigan and Dr. Brad Quo out of Mass General Hospital and Harvard. Now, these two are also members of our medical advisory board, and what we found is the medical advisory board is extremely important when you come from an engineering or tech background in order to understand what the customer really needs. Now, because I don't have a clinical background, I don't know what the clinics or how the clinics really operate. I don't know what the customer really needs. And so I need the input from people like on the screen in order to develop the right product for the use case. Now, as you can see, we have some of the most prestigious names within functional gastroenterology with people like Professor Eamon Quigley, who is a past president of ACG. We have, Professor Bill Shea from University of Michigan. Anyway, you can see these guys are top of the field. And as you can imagine, these sorts of KOLs don't need to be involved in projects like ours. We don't pay a lot of money because we can't, because we are an Australian-based company with not a lot of money. We've been doing this on the smell of an oily rag for quite some time as we're trying to get to market. But what we're doing is so interesting that these people are actively taking time out of the day to be involved with the research that we're doing, which gives us a real boost as engineers and scientists to know that we're going down the right path here. So what they've done is not only sort of helped us with our pivotal trial, but they've helped us flesh out what is quite a broad clinical program. Now, as I was mentioning earlier, we are creating what is generally a new field. So there's a whole host of work that we have to do in order to generate the data. So we have 2 core pillars of clinical indications that Atmo is going after as first and second priority. And as you can see, they're on the left there, which is around motility and following it up with small intestinal bacterial overgrowth. I mean, that's an obvious one for us. We measure gases through the GI tract. So why wouldn't we be applied to understanding overgrowth within a specific region of the GI tract? But to go beyond that, we are looking at IBS, we are looking at IBD, and understanding the interaction between the microbiota and the host through functional foods and through interventional studies, which is what is all on the right here. And these are investigators that are interested in the outputs that we have, contacting us really and wanting to be a part of our research. And in some cases, and as we're getting closer to market, actually paying for our devices to be a part of their research. So we've had over 1,100 capsules ingested within clinical trials, and they're in multiple regions: Australia, New Zealand, Asia, US, Europe. You know, we're kind of working across the board here, and with some pretty big names going around. So I kind of wanted to finish with this slide here, which is a bit of a sneak peek under the hood of what it takes to get a device like ours ready for regulatory submission. So we've had, as you can see on the left there, over 1,100 ingestions. We've had 4 different iterations of our device from when we first started the company back in 2019 to where we are now in 2024. And each time you get into the GI tract, your sensors shed a light on what's actually happening and where some additional gains might be made. So we've iterated our product as we get clinical data. So just always be ready to have your product driven by what your customer needs and but also what your data is showing you. Obviously in med tech, you can't iterate quickly because there's checks and balances. So you always have to operate within those QA systems, but it's important to reflect on the data that you're getting to improve your device. And so that's the journey that we've been on at Atmo to where we are submitting to the FDA at the moment. So thank you guys. Happy to take any questions you have.
Thanks, Tyler. It's excellent stuff. I've known Mal, chatted to him about this for years, but there's stuff now I had never seen or heard before. Questions from anybody? I've got one which just hit me when I saw one of your slides, but just to check myself there first. So you know where the capsule is across the whole system, right? And it's,
yeah, we only know when we go from one organ to the next. So I can't tell you where in the small bowel you are, but I can tell you when we've entered the small bowel and where we've exited the small bowel.
And the data's collected or transmitted in close to real time.
We used to. So in the earlier versions, we had it real time to the cloud, so we can monitor it as it was going. But what we learned from the gastroenterologists is that they actually really didn't care about that. So whilst we thought, oh, this would be a great improvement, we can do this, they did not care at all.
Didn't care.
Well, because they don't have time, right? A gastroenterologist sees a patient for 15 minutes.
True.
And that includes chart review beforehand and decision making afterwards, right? So within that 15 minutes, they're not going to take time out of the day later to review, oh, how's this test going in patient X?
Yep.
So
but look, your chart when you showed that, the different places with the pills, that it made me think. And now I don't know about therapies for different conditions, but it kind of makes it a great delivery tool for site-specific therapies, if there are any for that. Because up to now you didn't really, any sort of therapies become kind of whole body wide. But if you know where the capsule is and it was a vehicle for delivering a therapy to a particular part of, you now know that, but you didn't know that before. Like I said, I don't know much about therapies, but if there is a therapy or therapy developed that could be either reduce side effects or increase effectiveness because you can deliver it at a targeted site,
Yeah,
That
look,
would be great use of the technology.
absolutely something that's on our radar. There are a couple other devices that purport to do this, also still in development stage. So realistically, we do see our technology being able to be utilized in this fashion.
Great. Okay. Anyone else with any questions? There must be some after that. Please don't be shy.
Shall we?
Peter? Oh, can't hear you for some reason.
Can you hear me now?
Yep,
Yes.
absolutely. Great.
Okay, awesome. I just had one quick question. I was just wondering what sort of effectual change to the treatment after diagnosis you were envisioning based on the information that you gathered through this pill.
Yeah, it's a good question. So, well, just thinking about purely around motility here. Right now, diagnosis of, say, gastroparesis is done just understanding one organ. But what we show and what other trials have shown is that quite often that delayed gastric emptying is not driven by dysfunction within the stomach, but actually a dysfunction within the colon. And so purely from a systems engineering point of view, which I'm not a clinician, so as a systems engineering point of view, you delay the end of the tube, you're going to delay the top of the tube. And that's exactly what's happening in a lot of these cases, right? And so if you aren't able to understand or treat the delay lower, you're not going to fix the top end.
Yes, thanks.
But, no, Kent, if your question was really quick. Otherwise, if you could hold
just a really quick question about the hereditary pathway. So you mentioned that you did iterations as you went through the years and you conducted your studies. I was wondering how you factor that alongside your quality assurance path.
Yeah, look, it's tough, right? If you've ever worked under any sort of QMS, there are processes and procedures to do so. I mean, we're ISO 13485 accredited, so we do everything under our QMS processes. But because we aren't on market yet, it is still easier for us to make these iterations. I'm sure Greg could probably give you an understanding of what it's like to update your device once you get to market, because that's is another burden on yourself on top of what we're doing. But it's just paperwork, right? It's time, effort, and paperwork.
Thanks, Graham. Yeah, thanks very much for that, Kyle. Like that other question, we do have the panel session at the end where we can ask any of the speakers a question. So I'll hand over to you, Di, to introduce our last speaker.
Okay, so it is with great pleasure I get to introduce Greg O'Grady, who will be our next speaker. And Greg is a beacon, a shiny beacon in the New Zealand medtech innovation ecosystem. So he's a clinician, a bioengineer, and an innovator and an entrepreneur. And interestingly, Alimetry was also started around 2019. So Atmos and Alimetry are about the same age, actually similar ages. And it's really quite interesting to see the two companies being presented here side by side. You can also read Greg's very prepared bio in the agenda that Ella sent, and as Ella will tell you, I go rogue on her. But the one thing I'd like to point out is that Alimetry's values are around compassion, advancement, and trust, and I think that's fantastic. And I also think that that's the values that Greg himself embodies. So there you go, Greg. Over to you.
Thanks, Di. This has been a great session and really enjoying it. It's awesome to see the innovation coming out of Australasia and the gut. Certainly, motility has been a pretty hollow field for a number of decades, and so it's a pleasure to be sharing the stage with Atmo and Ken and Jerry and seeing the great work being done down here. I'm going to tell you about Elimetry. Some of you already very familiar with the company, others not. And I'll get through it relatively quickly so we can get to the questions you may have. So Elimetry, as I said, founded in 2019. And the name really stands for elementary telemetry, in that we develop wearable medical devices, wireless medical devices to diagnose gut disorders. And the field of problems we work in, sorry, I'll just get my slide order wrong here. Give me 1 second. The problems we work on are these really common symptoms of gastric distress. And they're shown here, sort of indigestion type symptoms, which are pain, burning, bloating, early fullness, excessive fullness. And then the more kind of really terrible ones for quality of life, which are nausea and vomiting. And at presently, clinically, we define these under these disorders: functional dyspepsia, nausea and vomiting, and gastroparesis. But it's fair to say the field is really confused about what's going on in these disorders, as I'll discuss. They're really common. The US burden is about 12% of the population have some form of indigestion seeking at least pharmacy level care. And 3% would have nausea for similar indications chronically. So extremely common and growing for reasons we don't really understand very well, but probably related to diet, viruses, stress, various other things, and society, leading to really a huge market. So the problem has been clinically is that we've really lacked reliable diagnostic tests for these patients. And that means that patients end up on this kind of diagnostic treadmill where they're just going round and round with inconclusive testing. They get frustrated and clinicians get confused, leading to trial and error care, and all based on symptom evaluations rather than biomarkers. And it takes on average 5 years for these patients to reach a diagnosis, doing multiple tests per year, and often getting excessive radiation. Our product has all been about trying to shorten this by, instead of doing a series of rule out steps to rule out diseases, instead locking in a specific positive diagnosis to get patients off this treadmill as quickly as possible. Ping has already talked about the stomach being this quite complex organ, where it has this part of the stomach which relaxes when you eat and then contracts again as you digest the food, and these big contraction waves going down in this valve at the bottom. And because it's quite a complex organ, currently the only way to measure it is through transit, which Atmo have talked about, which is one measure, but it's quite an integrated measure of what the stomach's doing. It measures the whole organ function, whereas we really know now that there's a number of different things that can drive these symptoms that patient experiences. The symptoms can come through the nerves and muscles, through hypersensitivity, or through dysbiosis and immune things, kind of what Jerry talked about for a while there, or the valve or the brain. Stress and anxiety can cause nausea as well. So there's all sorts of things that can influence it. And what we're trying to do is provide more specific diagnoses to some of these that can be used in addition to other tests like transit to reach a specific diagnosis. As Ping has said, one of the main things in the stomach is this underlying electrical activity. He talked about it at length, thank you, Ping. But it's got this pacemaker, and just like the heart, these electrical rhythms drive the contractions. And it's this that Elimetry primarily has been seeking to reach, which has been out of reach at the moment from the diagnostic panel that a clinician may have. So as Ping said, this has been prophesized for a long time that clinicians will be able to do this, but really it never came to clinical practice. People tried, but the approaches were really simplistic because they were using a small number of electrodes, often just 3, trying to mimic cardiology approaches. And their signals are quite different from the heart. They're 100 times weaker, and they suffer from very large amounts of artifacts. So to get them out reliably is really hard. And people's stomachs are also very variable anatomically, and different shapes, different BMIs can affect this as well. So it's a very hard problem and really didn't reach clinical grade. So Ping talked about the program that was going on in Auckland to map the stomach. And the first red exercise we did was to gather what the ground truth was in the stomach, to know what the stomach's actually doing in health and disease. And Ping talked about this. We were able to layer these electrodes on during surgery and see these electrical wavefronts, and then map patients with disease states where we could show abnormalities. And what you're seeing here is waves going normally down the stomach from top to bottom. And then you'll see them becoming very irregular and collide and scatter and do these kind of fibrillation type activities like the heart does. And we know that these disorganized patterns correlate more with when the cells and nerves in the stomach are damaged. And so we've got a biomarker that can help to target these neuromuscular disorders that currently can't be diagnosed. What the clinician wants is a way to differentiate when it's the stomach causing the symptoms as opposed to when the brain or the nerve or something else is doing it. So that's the biomarker we're going after. This was way too invasive for anything clinical. This was very much surgical research. So we then set about with Armin, our co-founder, came from San Diego, and he'd been working on these minimally invasive or non-invasive approaches to try and measure gastric function, initially with small wearables that he was building and kind of custom apps, but to show feasibility in this really important study in 2019 that he published in a journal. And at this time, we got excited about what he was doing. He was excited about what we were doing. And so we came together and we founded the company thinking this has got a lot of potential. We should build a product and take it out of the research space into the clinical world so that people can actually use it. So we started to build devices. This was Armin's starting point back in 2019. It was a mess of wires and a shoebox full of, and clearly not ready for clinical prime time, but this was kind of the level of electrodes we were needing to reliably pull out that weak signal that's 100 times weaker than the heart, to extract it from all the noise. We were summating it with many electrodes at once in order to pull that signal and show it. In order to make it more scalable, our kind of pandemic project was to build this array, and just like you can screen print t-shirts, Ping kind of touched a little bit on this too with the polymers. These are just traditional conductive inks filled with silver chloride that are screen printed onto plastics in order to build wearable sensors. And we really lead the world now in these high resolution flexible electronics for medical applications. And it's a non-trivial exercise to get these produced. We had to go through processes to manufacture them. This is just one step in the process where you can screen print these into reel-to-reel in order to make it scalable. And then the next step of pinning on the hydrogels and the adhesives and getting them packaged was even harder. But all problems that we had to solve along the way to get Elementary up and running. So the product is shown here. It consists of this wearable sensor pad with 66 electrodes on it, and this reader device, and we make this still within a production facility that we've set up within the university. So we make that ourselves in New Zealand here, and the sensor we make in the US. We've got apps that we develop alongside it, both to control the device, but also for patients to log their symptoms. And we've discovered this is just as important as the electrophysiology, getting robust symptom profiling alongside the test so that clinicians can look at the two side by side and correlate them. When certain things happen in the stomach, certain symptoms might arise, and we want to know the correlation between those two events. Very important to us, like Atmo, we have a cloud-based system, so we log and bank all our data, and we have a very active AI program. We're submitting our first AI regulatory submission this year to the FDA. We've had about to be four FDA clearances on this, so our strategy has been to go in with a relatively low bar and then regularly iterate to update the features of the product to, as we get customer feedback and product improvements developed, we add them to the product to regular FDA clearances. We've also got clearances in the UK and New Zealand, and we're in all three markets. Just to show you a test being done here, this is the system that's worn by a patient, and you'll see it's quite large. Because the stomach's very variable, we have a large field of electrodes to capture it. This is the reader device on the side of the patient. They come in fasted, they stop their motility medications that might affect the stomach, and then there's a skin prep step before we stick it on, and that all takes 5 to 10 minutes setup time. Then the patient sits comfortably in a chair, and they log their symptoms into our app during the test. And we feed them an off-the-shelf bar and drink in order to stress the stomach, make it work, and also to provoke symptoms shown here. And then so they consume that. And then they sit for 4 hours afterwards, which is how long the stomach takes to digest a meal, while they're logging their symptoms every 15 minutes. And currently it's all conducted in a clinic, but we certainly have scope in future to do it anywhere. But it's very easy to access. This is the doc that's cleaned for reuse at the end, and the report that's sent back to the clinician by the cloud. So it's really easy to access. All you need is an internet and a chair, and you can do the test anywhere, whereas a lot of other devices need to be done in large radiology suites in tertiary hospitals. So similar to Atmo, democratizing these technologies, getting them out into the real world. This is our symptom logging app, just to show you a bit closer. We have this dashboard display. It's all been highly validated as a patient-reported outcome tool, and so it's proving very useful. We've also now introduced a mental health screen. This went through the FDA late last year and got approved. So we have a validated proprietary mental health screen because we find a lot of these patients suffer anxiety, stress, and depression type symptoms. So that we included that as part of the test, and we continue to layer in these features that are done and can be seen by the clinician in the report. So what do we see from the test? This is when the waves are normal, shown down here. When these waves you saw earlier propagating down the stomach, we get a really stable rhythm. This is the high resolution electrogastrogram, it's called. And so we see the frequency here. They had a meal and the amplitude of the stomach went up and then it came back down. And that's shown by the brightness on this display and all very regular. So without understanding too much, you can see that this is all pretty regular and looks different from, say, this one, where things are really scattered. And compared to the last one, you saw these are all over the place. The stomach's very irregular. And we know that this patient had lost a lot of their interstitial cells that make these electrical rhythms. And so it's a bit of a bag of worms. And you see this very scattered trace. And so this patient has a neuromuscular profile, and that's a biomarker we're able to uniquely detect through this test is whether patients have this neuromuscular profile versus a normal looking stomach. We've got a reference range, which was really critical to getting this test in front of clinicians. So we've got 4 biomarkers we pull out in the reference range: the rhythm of the stomach, the frequency, things King talked about earlier, an amplitude that we adjust for BMI, and a fed to fasted ratio, which is how much the stomach responded after a meal. So the clinician gets these hopefully any day now. The FDA will approve us putting these on the report. They've already said they will. And so that'll be available to the clinician alongside the test for them to interpret the reports. Just to show how this is used in practice, this was a study we did last year, a bit earlier, on chronic nausea and vomiting patients, which is one of our main patient groups. And we took about 86 patients, actually half of them patients, half of them controls, and we put them through the test. And the important thing here was to show we were able to subgroup patients with nausea into those with abnormal stomachs and those with normal stomachs, but exactly the same symptoms. And so again, it's using this biomarker of neuromuscular disease with these irregular stomachs to separate out the subgroups on the patients and to show which ones have this more serious degenerative stomach disease versus these normal stomachs, and their symptoms are coming from somewhere else. Really important clinically because these patients need quite different treatment pathways, and at the moment, clinicians can't determine. The really interesting thing in this study was that our test correlated the biomarkers quite strongly with symptoms, which is what you want to see. But when they had normal tests, their symptoms correlated with health psychology. There are much higher rates of anxiety and depression in these patients in this study, which was, again, important to show these are separate subgroups that we can pick out. So something we're doing now as well is correlating these symptoms versus the amplitude of the stomach to show who has sensory motor disorder. This is another category. We have that neuromuscular category where things are very scattered. Now we want to know who has the hypersensitive stomach, so the stomachs their symptoms are coming from the stomach motor patterns. And so we've introduced into the report these correlation scores between the gastric amplitude, that's when the stomach's working, and the symptom logged at the same time on the app. And we can show these correlations, and other GI tests often do this. And so it's good for clinicians to see, yes, the symptoms are coming from the stomach. They have a normal stomach function, but they have maybe a hypersensitivity, which again needs a different form of treatment. And so kind of finally, the game we're playing with this is to put patients into these specific categories of disease. We build towards these phenotypes. We call them shown in white. And these already exist. We didn't invent them, but it's a chance for the clinician to, instead of just trial and error and guessing, they can now say with some confidence using the test, which one of these diagnostic categories the patient may fit into together with their other information clinically that they're receiving. And that leads to specific treatment pathways shown in blue. And so the test acts as a biomarker tool in order to put patients into these categories. And our clinical studies show that about 80% of the time we can yield an actionable biomarker, and about 75% of the time the test is impacting clinical care, which is really good for a diagnostic. So what's our value proposition with this product? We've done quite a few studies on this now and shown that we're generating good value for the payer, the patient, and the provider. In medical devices, it's a multi-sided market, and you have to think about all 3 of these categories in order to have a successful product. And so we're able to reduce the time to diagnostics. The test is safe. Patients love it because they're getting a lot of tubes put in them in the GI field, and so not having to do that and not having to be irradiated, they love that. And through this, we can reduce the needless tests and admissions that patients are having by getting them off that diagnostic treadmill, and it saves about $5,000 per patient per year when they have nausea. That's how intensively these patients are managed at the moment. And like Atmo, this leads to a greater revenue for GI doctors rather than sending them out of office. So thanks for inviting me. That's Limitry in short, and welcome any of your questions in the panel discussion.
Thanks, Greg. It's really fascinating to see how Limitry's journey has actually progressed over the last few years, and how more sophisticated your product and the thinking has been. So congrats on that. Okay, have we got any questions for Greg?
As always, I've got one, Di, and we can
Okay,
kind
you
of
can
move
have
into
one.
the panel. Is that I only ask this question because it's not my space, but in those more than 75% of cases where the clinical care changed, what are some examples of that?
Yeah, good question. So it's either a change in diagnosis or a change in management that we track for that metric. And these are things like a change from, I thought it was an actual stomach problem, to, oh, I've changed my mind and it's a nerve or a gut-brain problem. And then that leads to different treatment pathways, such as taking them off invasive therapies and putting them more towards the psychological and diet therapies, or putting them on certain medications. For example, some medications pump the stomach harder, others dull the symptoms coming from the nerves. And so the clinician's able to know more accurately which group they go into as a result of the test.
Oh, thank you.
So Greg, I've got a crazy question. So, I mean, like when the gut behaves wrongly, as you describe it as an arrhythmia, how do you reset it?
Yeah, great question, Di. So we don't have treatments at the moment for arrhythmic stomachs that are specific. Like cardiologists have a range of pills they can use to restore normal rhythms. And it depends what's happening in the patient. And a lot of these patients, imagine a building full of wires and you're trying to get electricity through it, and you go in and pull out bunches of wires and no current will flow through the building. It's a bit like that. The system is just so damaged that they're expressing these really abnormal patterns. So it's also more complex in cardiology in that resetting these rhythms is hard. But the important thing is knowing who is who at the moment. So for these patients with the really irregular stomachs, sometimes if they've got diabetes, we can reverse it. But a lot of them have these idiopathic immune states where their stomachs are constantly damaged. So at least we know who those patients are. We can treat them through more invasive symptom control, sometimes invasive neuromodulation devices, operations to bypass the valve of the stomach, those sort of things. Whereas the ones who just don't have that, we're able to take them out of that invasive pathway and put them on more neuromodulated drugs that might dampen the nerves, or together with psychologists, get them away from these kind of invasive treatment pathways. So while we don't have those perfect drugs at the moment, we're still able to separate the patients into quite different treatment streams.
Okay, thanks. I mean, it's always good to know that if there's a diagnosis, there's a therapy as well, right?
Exactly, yeah, crucial.
Di, can I ask a question of Greg, please? Greg, it's Faye here, and thank you for great presentation, exciting work you're doing, and really is inspiring. But I'm just wondering, I mean, this incredible technology you're applying to the stomach. I'm just wondering if you can replicate, or where your next stage is perhaps maybe the large bowel with irritable bowel syndrome, or is there other applications you're starting to look at for your technology?
Thanks, Faye. Yeah, good to hear from you. And absolutely is the answer. We have had a very active colonic program, and that includes irritable bowel syndrome and constipation, and we know we can do it. We've patented and published work showing that we can achieve these signals in the colon, and that's definitely the next target for us in terms of organs. It doubles our addressable market. There's so much going on in the colon, like the stomach. So in short answer, yes, but it will be a couple of years before those come through the pipes from development to product.
Thank you. Well, good luck.
Thanks.
I've got a question for any of you. I think what's been really cool about this workshop is we started with Jerry, people working and researching the roles of the microbiome, all the things that go wrong there. We've got people working on electrical activity and how that influences what goes on, and then people working on identifying and facilitating the identification of gases, or within the system, surely you guys are talking to each other about overlapping what you're doing, whether it's, you know, bringing together two areas of research on a common problem or something. What conversations are you having with each other?
Yeah,
in that sort of way.
Oh, yeah, it's a great question, Lance. So, yeah, we actually talk to each other quite a lot. The community for GI motility is quite small. There's not a lot of people working in GI motility. But the advantage of that is, you know, is everyone knows everyone, and sort of everyone's on board with the types of research, you know, people are working on. And it's sort of, you know, we're sort of finding out new information as we run these trials, you know, even just, you know, identifying novel sort of profiles and like the gas profiles in our patients. You know, as soon as we find something that's quite novel, we're always trying to figure out, you know, what do we do with this information, and, you know, what's the next set of experiments we can do. In fact, one of the experiments that we just did was we found these unique CO2 profiles in our gastroparesis patients, and we actually worked with Greg and with the Limetry to do a simultaneous study. So the patients ingested the Atmo capsule while they had the gastric mapping done, all at the same time. So we're not only looking at the gas profile, but we're also seeing, you know, what the gastric profiles, the gastric mapping profiles look like. So I think with this technology and with these sort of partnerships, we're able to do experiments like this to get as much information as we can to kind of answer those questions.
Yeah, look, to reiterate that, like, it's a small community. I know, Greg, I reach out to the Limetry guys all the time when I have questions. They do less so to me. I don't know if they just don't trust my opinion, but they've been through it before, so they have great advice for me. But I see us as complementary devices. I don't see us generally as competitors here. We're all trying to understand or unpack what is a confused field. You know, there's the patient outcomes are really poor here, so there's a use case for both of us together.
Yeah, that's exactly what I was thinking. I mean, that is excellent to know that that's going on, and hopefully there are people within the audience here now that are doing some things that can benefit or maybe complement on what they've heard today as well.
Can I take a different take, a slightly different take? And this question might be more for Greg and Carl. But, you know, in your journey so far, what's been that one thing that has really made, you know, your commercialization journey a success to date?
One thing making this hard, Di. Maybe 1,000 things. But yeah, I don't know. I think for us, the magic has been the team and just having really experienced people in all the right roles in the company. And we've been really lucky to have those people join the company and just have an awesome momentum and culture as well. So it's just absolutely the people that you're able to attract into these ventures and the contributions they make. Yeah, and the nice thing about New Zealand and probably Australia too, I guess, is the talent density is quite high. People are looking for good things to do. We have great ecosystem of science, engineers coming through especially. And so, yeah, I think that's pretty powerful.
Okay, thanks, Greg. That's
been our form.
Okay.
For us, I mean, yeah, I'm going to say the people as well, but I actually think the engagement with the general GI community, I think they feel like they've been in the motility space anyway, they've been ignored. And you go to these major conferences like DDW, and everything's about IBD. There's really nothing about motility. And so as soon as you come in with some innovation or you're trying to unpack a problem, they're all willing to help you out, right? Because they want outcomes as well. So I think clinician engagement has been a big one for us.
Okay. Thanks, Carl.
I think the answer you're looking for there is the existence of TTM and BioBridge workshop, right?
No, actually, it's not. Definitely not. But
since you
actually,
mentioned it,
Just saying. I like to
But
mention it just in case.
well, actually, I was going to say, I was going to ask you and Jerry, Payne, what successes for you both? What does success look like for you both? Because, you know, you're in the science field, it's a longer term play. And I know that you're working with like Greg, for example, but what does it look like for you?
I think pushing fundamental science into actual commercial and clinical applications is always key to actually have a end users would actually use the outcomes from science rather than that outcome being a publication. I mean, it's important, don't get me wrong, but at the end of the day, stuff you can get into people's hands and they can use, in my opinion, especially as an engineer, is probably more important, meaningful, and in a way, a good test of the robustness of the science than just a publication on its own. But that could be a controversial opinion.
Ask you say, don't let Marion hear that.
Yeah, no, I completely agree with Pang. I think, it's good to do research, but, what's even better is seeing that research being applied and kind of seeing that being integrated and kind of used by clinicians. For us, I think for me personally, success is sort of building that kind of workflow about finding where the problems are and then taking that back into the lab and then out into trials and then out into the market. And it's kind of, I've been sort of working in kind of this GI motility unit for 10 years. And I think over the 10 years, we've seen all these different components kind of come together, that setting up our own clinic and then setting up the patient's advocacy group and then setting up the clinical trial center and then setting up the commercialization pathway and kind of building all these different pieces. So as any, as innovation we create or, partnerships we have with industry, we're able to take that through that workflow. And I'm really hoping, maybe in the next 10 years time, I can kind of create this formula for this really good, pathway of finding unmet clinical needs and then really taking that right through into the market at the other end. Okay,
thanks, Jerry. I mean, I'm also fascinated by the fact that, in what you've built, you haven't forgotten the patients. And so with the patient advocacy group, just closing the loop.
Well, it's really important having the patients on site because we couldn't do those trials with Greg or Kyle if we didn't have those patients. Because, as soon as you put up that kind of information about, here's gastric mapping or here's this capsule, we get emails, we get flooded with emails about people who want to participate. And it's like what Carl's saying, that's been sort of instrumental for us to get those recruitment numbers and to get things, off the ground as quick as we can. It's something we provide to the patients, in terms of support and the network, but it's also, we get 10 times the value back from the patients as well, because they kind of help us out so with these trials and fundraising for research as well.
Yeah, that's cool. It sounds like you need to work with Payne and set something up here too, on this side of the Tasman, right?
Yeah.
Yeah, cool. Right. Any other questions?
I was going to make a comment on Jerry's just noting Ping's comment. He needs to leave soon. Your earlier words, Jerry, I agree. I kind of spend a lot of my time in the commercialization space, and sometimes researchers think I'm kind of anti-research, but it's not either or. We need both. We need the blue sky research because behind all those value delivered and technology developments, someone did some research and got some outcomes that people didn't know about, and that was people turning those outcomes into value and heading down the commercialization path. So we need the blue sky research. We need that kind of feeding the delivery of value of those things. So it's never either or.
Yeah, I think you got to take that risk, and with basic research, so you get that room to take risk and kind of try and sort of find new things. And it's some of the work we're doing with the fecal transplant research. It's relatively new, it's relatively sort of blue sky work, and it's really interesting, but I think it's quite far from commercialization in that aspect.
Yeah, you definitely need to, you know, without that pipeline, you really have nothing at the end of the day. Okay, any other questions? Because Peng's about to go.
Oh, I'm actually fine now. I don't need to leave. So good.
Oh, okay. But in any case, any other last comments? If not, we will return to you a few more minutes of your time.
Maybe I'll just say a quick how impressive it is to see Australasia leading the field here, because there's not much happening overseas. So the depth of the presentations that you've seen today hopefully show that we're leading the world down under here. So it's a really positive field for the country and region. Thanks for everyone who's contributed to that.
Yeah, I was going to say, I think Australia and New Zealand do a lot of really good things, and it's just kind of hidden, just under the surface a lot of times, but we should really be better about talking about ourselves in the future. But yes, you guys are fantastic, all of you. And thank you so much for such an inspiring session. And maybe to say thank you and wish and to close the session, maybe everybody could just put their cameras on so that our speakers can see the people that are here.
Think of like that all these people who work in gut health are really a shy community. I would have thought the opposite.
Yeah, that's cool. So thank you, all four of you, and thank you for giving your time to the BioBridge initiative.
Yeah, thank you so much for that, and thanks for everyone joining and staying to the end as well. Good to have you here. Yeah, thanks very much, everybody.
Thank you all.
Thank you so much, everyone, for listening.
Bye.
Bye.